Neuroscience and beyond. Know more, get inspired. Welcome to the Neuroscience and Beyond podcast. My name is Viljan Georgiev and I am a PhD student at the University Medical Center Göttingen in Germany. Together with my PhD colleagues Christina, Sabina and Lenny, we desire to bring science, and especially neuroscience, closer to the general public. In the following episodes, we will talk to various scientists about their inspiration to do science, their research topics, and their opinion on current scientific problems. Today, our guest is Professor Tiago Outeiro. Professor Outeiro is a director of the Department of Experimental Neurodegeneration in Göttingen, Germany, and a full professor at the Institute of Neuroscience at the Newcastle University in the United Kingdom. His research focuses on the understanding of the molecular mechanisms which lead to neurodegeneration in diseases such as Parkinson's, Huntington's, or Alzheimer's disease. To address these problems, Professor Outeiro and his team employ a wide variety of model organisms, from the simple budding yeast to mammalian cell culture and mice. The ultimate goal of Professor Outeiro's lab is to develop novel therapeutic approaches for these and other related disorders. His team works closely together with clinicians in order to accelerate drug discovery and potentially translating basic research into clinical applications that will improve the lives of patients. Before we begin, I'd like to mention that this podcast is separate from my research position at the University Medical Center Göttingen. This podcast is supported by the International Max Planck Research School for Neurosciences in Göttingen, as well as by the Cluster of Excellence Multiscale Bioimaging in Göttingen. And now, please enjoy the talk with Professor Tiago Outeiro. We would like to apologize for the technical problem that we experienced at the beginning of the podcast. Professor Outeiro, thank you for being here. It's a pleasure to have you here, and I'm looking forward to our discussion about science and about your history behind the scientist you are today.
No, thank you very much for the invitation. It's a pleasure and an honor. I like podcasts. I listen to them on my way to work or on my way home, and so I sympathize a lot with the cause. I think it's a great way for promoting knowledge or information, and so I'm very thankful for the invitation.
Great. Then it's going to be wonderful discussion. So far, we have started by asking our guests what inspired them to become scientists. Could you please briefly summarize your story?
Yeah, absolutely. So when I was a student in secondary school in Portugal, I wasn't really sure what I wanted to do. I knew I liked sciences, but I happened to also be good at the time in math, and so economics would also be something that could be interesting. But I liked science. I liked biology and chemistry. And as I went up in my studies, at some point I had to face the decision: what should I study? Where should I go to college, and what should I study? And then I wasn't really sure. I felt I didn't want to be a medical doctor. I don't think I would be good at dealing with human suffering and several of the aspects of medicine. And so I knew I kind of didn't want to do that. And the other possibilities would be to go more into biology, biochemistry. And at the time in Portugal, the degree in biochemistry in my university had been already running a few years, and I knew that it had good reputation. So I decided, well, maybe this is a possibility, maybe this is a path I can follow, and then see what this brings me to. I confess I didn't have a very good idea what I wanted to then specialize on. And then it was actually on my third year in college that in Europe, in the second half of the 90s, we were going through this big scare of the mad cow disease pandemic. Everyone was afraid of this disease that was different than what people were used to, and everyone was thinking that we had to be very careful with eating meat because maybe the disease could be transmitted into humans. And as a student, as a third-year student in biochemistry, I was reading the papers, and I was reading about the seminal work by Stanley Prusiner, who then got the Nobel Prize for his work in prion diseases. And I was reading also a lot about a phenomenon that was similar to the one that takes place in the human or the mammalian prion diseases, but that happens in yeast. There was an author that I was reading a lot about. Her name was Susan Lindquist, and she was publishing in great papers in Science and Nature and PNAS and all these top journals. And I was really impressed by that. But of course, I was a student. I was very far from thinking that one day I would work on this. And then I had this curiosity of going abroad to have an experience in another country. And since I enjoyed speaking English at the time, I thought, well, going to the UK would be a good option also to improve my English. And I ended up going to the University of Leeds, to the lab of a famous biochemist at the time, Professor John Findlay, who worked in membrane proteins, GPCRs, and lots of membrane proteins also in the vacuole. So I was lucky to work with some postdocs, senior postdocs in the group that worked with yeast, and I learned a lot of molecular biology. This was really cool. During those 7 months I spent in England, in Leeds, I learned a lot of molecular biology. And I ended up applying actually for a PhD in Leeds. But at the same time, as a student that, didn't know still very well what to do, I wrote this Professor Susan Lindquist, and I asked her, would there be a chance for me to come and work with you? I've followed your work for quite a while, and I admire what you do very much. And surprisingly, she wrote back, the next day, or I don't know, but it was quick. And I was, wow, this lady is actually responding to me. So I was really shocked because, you think that these famous people don't have time to respond, but she actually responded very quickly. And she said, yeah, well, I take good students. So if you have good letters of recommendation, maybe we can think about it. And one of my professors in college, she's an expert in a human disease called familial amyloid polyneuropathy. This is a disease that happens to be frequent in Portugal, in the north of Portugal. There's a region where mutations in this protein called transthyretin exist at higher levels because there's, these communities, they live together, and so they keep the mutations. And she was one of the pioneers in this field. So she identified some of the mutations. She had done a lot of this seminal work. And we thought that maybe we could apply some of the knowledge from these prion diseases, where at the time people were showing that by manipulating the levels of certain chaperones, you could interfere with protein aggregation. So we thought that maybe we could design a project involving the three of us. So I would be the student, Susan Lindquist would be the host, and Professor Maria João Saraiva would be also a co-supervisor, and we would try to study the folding and aggregation of this protein in the models that Susan Lindquist had in her lab in Chicago. And that's how it started. I eventually was very interested in this field of protein aggregation diseases. And I went to the US and I started my PhD. And actually, in the beginning, I was working on this protein transthyretin. We were developing new models. But eventually, we thought, this is probably not working so well after 6 or 7 months. And that's when, sometimes you have to make decisions. And sometimes the best decision is that you need to switch projects and to find something that may be more likely to succeed. And she had the vision to advise me to do that. Mm-hmm. And this was in 1999. So just a year and a half before, this protein that I now study a lot, alpha-synuclein, had been associated with familial forms of Parkinson's disease, and researchers had discovered that it's one of the major components of Lewy bodies. These are the protein aggregates that accumulate in the brains of patients with Parkinson's disease. And so we thought, well, this protein is new. This is a protein that forms in aggregates or protein inclusions inside cells. So let's study this protein instead. And yeah, the rest we can cover in in more detail if you want. But that's how it all started.
Yeah, but again, it shows some kind of a brave decision from yourself to write this person just out of the blue, as we say, right? Can you accommodate me at your lab? Do you have space? And this is a very good advice for young people to just try to contact the PIs that they admire, and maybe they will have a chance, as in your case, to start their career in this particular lab.
Absolutely. I think this is great advice. I always tell this to my students and to students I meet and that ask me, oh, so what should we do? Always go and don't be afraid to contact people. Don't be shy. If they respond, that's great. You get an answer. If they don't respond, then, they're too busy. They have other things, and don't take it personally. Just move on to the next person. And Susan actually always used to say something that I take with me, and I try to pass it to my students, and that's, the squeaky wheel gets the oil. So that wheel that is making some noise, that's where you're going to go and put the oil first. And that means you have to, not to be annoying, right? No one wants to be annoying and to just be always bugging people, but you have to make yourself heard when you need, and then you will get attention, and sometimes things work well. And I was lucky in that sense.
Exactly. I completely support this anecdote, or I don't know, how do you call it? This is really, really the case. And this is what we try to do here, try to increase the voice, and let's see if we will be heard or not about some scientific problems or opportunities. But to this point, we will go a little bit later. So you mentioned here several terms that we are going to clarify throughout our discussion, like aggregates, chaperones, some protein names, etc. Before we go there, I would like to ask you about maybe the second stop in the USA throughout your career. So, as long as I'm informed, you were involved in a biotechnological company. So you had some positions in the top-notch institutions such as MIT, Harvard, right? And your work in the lab was somehow transferred to the company. Could you please tell us more about this story? It's very interesting to hear more about it.
I feel I was very lucky, I had the opportunity of working in 3 major universities in the US. So I started at the University of Chicago. Then when I was kind of in the middle of my PhD, my supervisor moved the lab to MIT. And at the time, of course, I wouldn't be finished. I had to move, or I could have considered switching labs and doing something else. But I was really enjoying the project, and she gave all of us at the time the opportunity to move with her to Boston. So I knew, this could cause some delays in the project because we would have to start a new lab from scratch. And so, I was predicting some of that, but I really wanted to do it. And I had the opportunity at the time to be one of the first people to move from Chicago to Boston. So that meant I was there before most people were there, and at a time when we were buying a lot of the instruments and making decisions on what microscopes, what lab instruments we would get. And I was one of the people involved in calling the companies, dealing with the sales reps, and I was negotiating with them and, trying to get the best deals for the lab. So that was great experience. Then towards, in the second half of my PhD, we felt we had something that was interesting. Some of the findings we were having that we can come back to later were looking pretty interesting. And at MIT, there's this culture that if you have something interesting, then try to patent it. You need to protect it so that that you have a chance to develop things further. And that's something we did. And then there's this culture of entrepreneurship. And we hear about entrepreneurship all the time, and how important it is to try to start a company when you have something that could have an impact in human lives. And she was trying to do that. And eventually she connected with a colleague from California, from Scripps, a colleague that was also working a lot on protein aggregation and transthyretin, as it turns out. And so the two of them decided to start a company at some point. So they went to venture capitalists and they did fundraising. And at the time, I was still a PhD student, and she was involving me and asking me to come with her to meetings and presenting the work. So instead of being her presenting, I was there also presenting the work. And it was a bit intimidating. I was talking to all these, important people, and I was just a PhD student. But that meant that I was involved from the beginning. And so in this company they started, FoldRx, they wanted one side of the company was to use the models that I had developed during my PhD to try to do drug discovery for Parkinson's disease and other diseases. And we agreed that at the end of my PhD, I knew I didn't want to go and work for industry. I knew I wanted to do a postdoc and continue my academic career. But I thought it was a great opportunity to spend some time working at a company, learn even more how it is to be on that side. Mm-hmm. And so that's what I did. So I was consulting for the company. Then when I finished my PhD, I spent a few months working at the company in the lab, transferring the knowledge and the things I had done to the company. And then at some point, that was it. I knew I wanted to do my postdoc. So then I moved to another university in the Boston area, Harvard Medical School. So I was at MGH, and that was, again, a fantastic experience, a great place to be with lots of top scientists in the field. And I happened to work with another person that is very well known in the field, Brad Hyman. He's a leader in the field of Alzheimer's disease and Parkinson's disease. So I was reading a lot of the work that they were doing at the time, and being in Boston also facilitated things because I could talk to him. I met him, and we agreed that, he was also interested in someone with my background, and I eventually came to work with him. So that's how I went from MIT, from the Whitehead Institute, to the company, FoldRx, and then back to the university, to Harvard.
But that is a very unique perspective. You already had at this level of postdoc experience in the industry and academia. And I think this is nowadays, personally, I would like to experience something similar just to see how these both worlds operate and maybe combine them, take the most the most beneficial parts. But it's hard, especially here in Göttingen, for example. There are not that many startups, biotech. There are several companies, but they do some particular things, and it's hard to perhaps translate. Now, I don't know, with Fraunhofer Institute, maybe this will improve, but it is definitely different. Yeah. And maybe now it's a time to ask you what brought you back to Europe, and especially Göttingen.
Mm-hmm. Yeah, no, I agree. I think still on the point of biotech, Göttingen, I think it has great science, as we all know, great institutions, but the culture, the entrepreneurial culture in Göttingen, I think is still not at the level where it can be. Mm-hmm. For many years, scientists that were starting company were not seen very favorably by their peers. I think this is changing and has changed a lot in the US. This is not like that anymore. It hasn't been for a long time. But in Europe, things happen usually a little later than in the US. And so here in Europe, the culture of having someone that is an academic and that dedicates time to something that is for profit. The goal of a company is to make money. Exactly. That's the point, and to contribute to development. And that was not seen very favorably. People would think, you just want to make money. But that's not the point. You just want to develop certain ideas. And sometimes the best environment to do that is really within the framework of a company. And so Fraunhofer being here, and also with Sartorius promoting the life science campus and other opportunities that are now starting to be available. I think these things will develop, but we need this culture to be promoted. So, so after my postdoc, I felt I had personal reason to want to go to Portugal. So I had been away from Portugal for 9 years at the time, and I thought, okay, if I stay in the US for my next position, that would be as an assistant professor in a university, I felt, okay, maybe I have a chance of getting a job. I didn't apply, but I thought, looking at my colleagues that were getting positions in academia, I thought that should be possible. Not at MIT or Harvard, but I could probably get a job somewhere in the US and start my career. But those positions would mean that I would need to invest a lot of time for 5, 6, 7 years in a tenure track position to then have a permanent position. And I thought, okay, if I spend all that time here now, I probably will never go back to Europe, because then I would probably have family and have stronger roots in the US. And I thought, no, now is the time. And at that time, actually, I had been exposed to other people in Portugal through meetings and things that I had done in Portugal. And I was looking around and talking to people, and there were a couple of places that were interested in having me come there as a scientist. So eventually I ended up deciding to go to Lisbon, to the Institute of Molecular Medicine. That's where I started my lab in 2007. And that was a wonderful experience as well. This institute is one of the top institutions in Portugal. Of course, we know when we move from Harvard, from a well-funded lab, to almost any other country where you have to be your own boss and bring your own money, it will not be the same. But this place is terrific. I have to say, it's fantastic. It's very well equipped. It was run at the time by a colleague that had been at the MBL in Heidelberg, that had a lot of international exposure. One of the main persons leading this institution as well was a very famous neurosurgeon in Portugal that was very well connected with politicians and all the people that could support this type of institute. And so I felt I was in a good place. Fortunately, there's several other good places in Portugal, but I was in one of the good places. And everything was going really well. I was happy being back home. I also had a girlfriend at the time when I was in the US, and that was also a motivation to go back to Portugal. So everything was going really well until one time I was attending a Society for Neuroscience meeting in the US. And I was there, I think with one of my students. I had a poster, or I had a poster, I don't know. And a colleague approached me, and that colleague was Professor Matthias Baer from Göttingen. And he was, oh, hi, Tiago, I'm Matthias. We would like to invite you to come and visit. We have a position open. Okay, great. Sounds very, very nice. And we left it at that. And the truth is that a week or 2 weeks later, he wrote me an email saying that there was a position in Göttingen, a full professor position, and that they wanted me to apply. And I had just started my lab 3 years before. I hadn't done almost anything relevant. Things were starting to come out. And I really didn't think I was at that level to become a full professor. At least in Portugal, this would take me another 5 or 10 years. And here they were offering me a position like this. And I was really confused because I had just moved back to Portugal and I didn't know if I wanted to leave Portugal again. I mean, I didn't speak German. I didn't have any connection with Germany. I didn't know Göttingen. So I told him, yeah, thank you, Matthias. That's very kind. But, I think I'm not at that level yet. So, yeah, I will not apply. But then he invited me to come and give a talk, and I came. It was great. I had meetings with a lot of the colleagues here, and I really enjoyed. And at the end of the day, after meeting everyone, he said, well, we really think that what you're doing fits this position and the interests of Göttingen at that particular time, and we would like to offer you the position. And then I really had to think about it seriously, right? Then I had it in front of me, and so after thinking about it, and discussing it at home, I thought, well, this is a great opportunity. And I even talked to the dean of the medical school in Lisbon at the time, and he said, oh, we like you very much. We want you to stay, but you're too young. I cannot give you a full professor position. And this for me was, made everything clear. So for Portugal, I'm too young. Mm-hmm. But for Göttingen, which is an institution that is much better known, I'm not too young to become full professor. So then it was clear to me that I had to take this position because I didn't know when I would ever get a similar position in Portugal. And so I took the position, and we're almost 13 years later, and here I am still and enjoying Göttingen and the colleagues. It has been a wonderful experience, but we can explore that in more detail if you want.
Exactly. And I think, first of all, this is a really wonderful experience. And actually, we've heard similar story. I think one of our previous guests also was kind of a junior in his field, but he was recruited while at the conference on a poster by at this institution, as long as I remember. This is very interesting. Maybe there was some kind of agenda in Göttingen. Okay, we have to really recruit good people so that we can push science here. I don't know. Anyhow, you're here, as I said, 13 years in, and now you're doing a very, very, very great research. Can you please start telling us what is your research about?
Yeah. So, as I mentioned in the beginning, what brought me into this field was this interest for these diseases that are called prion diseases. These are very interesting and special diseases because they can be sporadic, so they can appear for reasons we still don't know. They can be hereditary, so that means you can have mutations in the prion protein, and this can cause familial forms of the disease. And they can be infectious. And this is what is or was so surprising. Being infectious meant that animals can consume contaminated products and develop the disease. Or you can have surgery, and if the surgical material was contaminated with this infectious agent, then these people would come down with the disease. And these diseases are terrible. They lead to dementia, and they kill you within months or not a very long time normally. And what was so special is that these diseases were not explained by any of the conventional infectious agents that were known at the time, bacteria or viruses or parasites. So using UV that destroys DNA, you could not get rid of the infectivity. So then Stanley Prusiner, the Nobel Prize winner in 1996, I believe, he proposed that the infectious agent was a protein, and not a protein that came from another organism. It's one of our own proteins. So it's the prion protein that exists in mammals and also in humans, in our brains and in other cells as well. And this protein can exist in at least in two, let's make it simple, in two different states. One that is folded normally, that makes the protein function at the membrane, that's where it's located, and one that is abnormally folded, that forms aggregates, so protein clumps that accumulate inside cells and in some cases also outside cells. And this process of how a protein goes from this normal folded state into an abnormally folded state that is aggregated and that we still think may cause some cytotoxicity was what was very interesting for me. So from then, I started working on that other protein I mentioned, transthyretin, and eventually switched projects and started working on alpha-synuclein. And what's common among all of these proteins and many others is that all of these proteins can switch their conformation and accumulate as protein aggregates. And alpha-synuclein, as I mentioned, was a protein that had been discovered just around 2 years before I started working on it. In the context of disease. I mean, the protein was known before, but it was actually being studied as a protein that was involved in the process of song learning by some species of birds. And so it was being studied in a very distinct context. And then researchers in the UK found that this protein is the main component of the protein inclusions found in the brains of patients with Parkinson's. And I wanted to understand how this protein is causing problems to cells. So the first approach was to use the models that Susan Lindquist was really pioneering at the time, which was to use yeast cells, the same cells that we use for making bread or beer or wine, very simple unicellular organisms, but are very well studied and had been studied for many decades in the labs. And so it was actually the first organism to have its genome fully sequenced. Mm-hmm. So we had a lot of tools available for manipulating yeast and for manipulating the proteins that regulate protein folding in yeast. Mm-hmm. And so we thought, okay, let's put this protein in this very simple cell. This protein does not exist in yeast. But we thought that a yeast cell is still a cell. So the main folding machinery in a yeast cell is not so different from the folding machinery in a human cell or in a human neuron. Of course, the cell is specialized to do other things. There's many differences. It's, of course, a lot simpler than a human neuron. But from the basic perspective of cell biology, a lot of the processes are highly conserved from yeast all the way to humans. So we thought this could be a nice model, a nice tool to study what factors in the cell govern the folding, the misfolding, and the toxicity of this protein. And actually, we found something that was, at the time, very surprising, which was that if you express this protein, you can see it at low levels that it distributes and it associates with the plasma membrane. And there were lots of studies coming out showing that this protein interacts with membranes. On the other hand, if you overexpress this protein a bit above the levels that the cells can normally tolerate, then the protein started to accumulate in clumps that we could see using GFP fusions and microscopy. And this was also causing toxicity to yeast cells. And this was, so we were working on this the same year that a study was published showing that in humans, if they have multiplications of the gene that encodes for this protein, alpha-synuclein, they get Parkinson's. So it was telling us, in humans, if you produce too much protein, this is bad. Mm-hmm. And we were seeing in yeast that if you produce too much protein, this is bad. So this was, for us, it was like magic. We were finding something in such a different organism that was perhaps related to what happens in humans. Mm-hmm. And we just went deeper into this. We took advantage of the yeast toolbox that we normally refer to as all the genetic tools and strains and collections that are available, to do some screens and to go deeper into understanding what pathways are affected by this protein. And that's how I started. And from then on, I continued working on this protein. I expanded and have worked on other proteins as well, like huntingtin that causes Huntington's disease, tau that is involved in Alzheimer's disease and other tauopathies, SOD1, which is a protein that also aggregates in amyotrophic lateral sclerosis. So we look at these proteins as models to try to understand how cells deal with the accumulation of these proteins when they somehow misfold, change their conformation, and accumulate as protein clumps inside the cell.
Mm-hmm, mm-hmm. Very remarkable that you at the time discovered this in this, as you said, simple organism, the yeast, and then at the same time humans show the same phenotype. This is just incredible. And I agree, this is a great model organism. As you have said, there is a tool, as with C. elegans, for example, or Drosophila, there is a huge tool that you can use and study these processes. But of course, if you want to go to drug development, you would need higher animals to test this, right? But before we go to this, maybe can you please summarize the function of alpha-synuclein in health before we start with Parkinson, and what is actually Parkinson, how it occurs, and how can we prevent ourselves somehow, if it's possible?
So alpha-synuclein is a protein that was initially identified in the synapse and in the nucleus. Then the name synuclein comes from the fact that it was found in these two compartments. Then it was called alpha because soon after they found beta and gamma. So these are proteins that are very similar to alpha-synuclein. They differ, one more on the middle region, one more on the C-terminal region, but they belong to the same family. So there's three synucleins. Alpha-synuclein is, of course, the one that is most studied because it's the one that we know is genetically linked to Parkinson's disease, and that attracts a lot of attention. What we know about its function comes actually from the work of famous scientists for Göttingen, like Tom Sudoff. He has done a lot of work on this protein, and other colleagues that worked with him and are continuing to work on this topic. And what we think is that this protein interacts with membranes not in a conventional way. It's not a typical protein that spans membranes, but it binds laterally to membranes, and it's thought that it could be important for maintaining membrane curvature, in particular of synaptic vesicles, and that this could be one of the functions of the protein to regulate synaptic vesicle trafficking and biology, at least in neuronal cells. Now, those are ideas that are backed up by a lot of nice literature, but this is only part of the story, because this protein is not only abundant in the brain, where it's thought to account for almost 1% of total protein, so that makes a huge percentage of total protein in the brain, but it exists also in red blood cells and in platelets, in other tissues. And in the context of Parkinson's, no one is studying what this protein does in red blood cells. And these cells, as we know, they don't have synaptic vesicles, so it's not there doing the same thing that it's doing in neurons. So this means we don't fully understand the function of the protein. We have these ideas that it may be involved in synaptic vesicle biology, but what it does in other cells and in other tissues is still not clear. And there's some recent data that is fascinating. We still don't understand it, but suggests that it may be actually important for us to help cells respond to infection by bacteria and eventually by, or possibly also by viruses, in ways we don't understand. But it seems that it may help the immune system deal with infections. And this is something that is being studied at the moment by several groups. So I think in the coming years we'll learn more about the function of the protein. And the idea is that we tend to be very simplistic and think one protein, one function, but biology tells us that one protein may play different roles, may have different functions in different cells, in different tissues. And so this is something we need to be considering.
Yeah, in terms of evolution, maybe it makes sense if you use one protein for different purposes instead of producing another protein and thus consuming more energy. If we think as a cell, right? I don't know. Maybe someone looked into this, and there is other cases. Perhaps there are many other cases of different other proteins. But then we have this alpha-synuclein, which tends to aggregate in some cases. As you said, there is some genetic aspect of this disease. And what happens to the brain when this protein starts aggregating, accumulating, and where it's accumulating actually?
So Lewy bodies, these protein inclusions, have been known since 1912, when the German scientist, I don't know if he was a neuropathologist, I forget now, but his name was Friedrich Lewy, working with Alois Alzheimer and the team in Munich. He identified in post-mortem tissue, brain tissue from Parkinson's patients, he found these inclusions that then a few years later were called Lewy bodies. And at the time, we didn't have antibodies. We didn't know what the proteins where we just knew it reacted in a specific way with some dyes that they were using at the time. But we knew those structures existed. But only in 1997, with antibodies, a colleague, Maria Grazia Spilantini, was able to find that those Lewy body structures stain positive for alpha-synuclein. So how this happens is still a mystery. What we know is that it seems to be an age-associated process. So Parkinson's disease usually, in most cases, doesn't appear early in life. It appears after 60, 65 years of age, although you have junior and early onset cases as well, in people around 30 years old, 35, 40, and you have juvenile forms that can happen in people that are 12, 15. Those are familial forms normally, those juvenile forms associated with mutations in other genes. So we think maybe the pathological mechanisms might be a little different. But in all the other forms, or all the other cases, or most, I should say most, not all, most cases have Lewy body pathology in the brain. So they have these protein inclusions. And we don't know why this happens. There's many possibilities. One is that some cells, for some reason, start overproducing the protein, and because this is a protein that normally is what we call intrinsically disordered protein. This means that under physiological conditions, the structure seems to be disordered. So the protein is not nicely folded as other proteins. It seems to be disordered, and this seems to be one important characteristic of the protein to enable it to perform different functions, to interact with different partners, different proteins. But what happens is that at some point, then this protein starts to misfold and to form aggregates, what we call aggregates, these protein clumps. Eventually, we think this is a continuous process, and you start with a small aggregate, and somehow it starts to grow, and at some point, it forms the big protein clump that we can then see under the microscope, and we recognize it looks like a Lewy body. But we think, and this is still debatable, but the evidence from all the studies in animal models and in cell models suggests that this protein aggregation can be a bad thing. It can cause toxicity. Now, there's colleagues now that are questioning this hypothesis and that say maybe what we have is not toxicity coming from the accumulation of the protein, but coming from the loss of normal function. Because if instead of having the protein in its unfolded or partially folded form, if now you have the protein clumping in one part of the cell, that means you don't have enough protein available in other parts of the cell to perform the normal function. And so this is now an hypothesis that is being tested as well. So is this a proteinopenia disease where you have problems because you don't have enough functional protein, or is it a proteinopathy where you have problems because the protein is causing toxicity? And probably in the end, it's going to be something in between. It's not just one or the other. I think it's going to be something in between. And we know from studies in animals and in cells that you can, under certain conditions, induce toxicity if you overexpress, if you cause this protein to aggregate. So we know this happens. We are just not very good still at inducing the formation of these inclusions, because in animal models, we and many others use transgenic mice or rats, even non-human primates have been used. And you've never seen, no one has ever seen something that you can say, oh, this is a Lewy body formed in the brain of a rat or a mouse or a monkey. No one has ever been able to do that. And we don't know if this is because these animals don't live long enough. We know, as I mentioned, the disease appears normally late in life. So it means that you need time for these structures to form and maybe to have all the components that make them look like a Lewy body. And in these animal models that live months or years, but not 60 years, maybe we just never reach the point where we can mimic this aspect of the disease. So there's a lot of open questions still, but we have evolved a lot in our understanding of the basic biology of the protein and cellular pathways that can be modulated to affect the aggregation and toxicity of alpha-synuclein.
Mm-hmm. I would come back to that point maybe about how do we utilize animal models to study this disease, although they don't show per se these Lewy bodies.
Mm-hmm.
But let's go back to Parkinson's disease. And so now we have these protein aggregates, and they happen to occur in some area of the brain that is damaged. And we know that kind of the initial part of neurodegeneration occurs in this particular area in the brain and then spreads around. Could you please elaborate on this,
mm-hmm.
this topic?
Yeah. So this knowledge comes from the work of several colleagues in the field, and we know that when people with Parkinson's disease seek help, it's when they feel the motor problems.
Mm-hmm.
They feel the typical bradykinesia, which is the slowness in movement, the resting tremor. It's not the tremor when people drink coffee. Oh, I had too much coffee, I'm shaking. That's not the
Mm-hmm.
tremor of Parkinson's. It's a resting tremor. It means that you can be with your arms resting or your legs, and you have usually one
Mm-hmm.
side that is shaking at a specific frequency. And you have also postural instability, so a curved posture, and muscular rigidity. So these are the typical symptoms of disease. So when people start feeling these problems, they seek a doctor, and that's usually when diagnostic takes place.
Mm-hmm.
Now, we know, or clinicians know, when they started looking at a lot of patients, that years before the onset of the motor problems, patients already have hyposmia or loss of sense of smell.
Mm-hmm.
They have often constipation, so gastrointestinal
Mm-hmm.
disturbances, and they also often exhibit REM sleep behavior disorder. So they have problems during their sleep, during the REM sleep, when you have the rapid eye movement phase of sleep. These patients they enact their dreams. So they are dreaming, and usually they are violent dreams. So they are punching and kicking, and usually the partner suffers because they get punched and kicked involuntarily. Of course,
Mm-hmm.
they don't want to do it, but these dreams are very vivid, and so the people are enacting the dreams. And sometimes you see videos in studies where people fall off the bed,
Mm-hmm.
because it's such a violent sleep that people fall off the bed, and that can be dangerous. And so this knowledge tells us that perhaps other regions in the brain are affected even before our brain shows the motor problems. And then another German scientist, Heiko Brack, is a neuroanatomist that has been in Ulm for some time now. He was in Frankfurt, but in the past years he has been in Ulm. He studied lots of brains from people with Parkinson's, and he did these Lewy body stainings to see where are the Lewy bodies in people that have Parkinson's disease, but with different severities.
Mm-hmm.
And he was able to find a correlation between disease duration and disease progression and severity with the load of alpha-synuclein in Lewy bodies. And so it seemed that people that didn't have symptoms, they had pathology mostly in the brainstem, in the medulla oblongata. And then as disease progressed, at some point, you could see Lewy body pathology in the substantia nigra. And this is when the motor problems normally arise. And then as disease progresses, you see pathology moving upwards in the brain, eventually reaching the cortex and other areas. And that's usually when you have signs of dementia and other severe problems. So this was the idea that perhaps as disease progresses, the pathology also progresses in the brain. And then in 2008, there were two very interesting studies, one from a Swedish group and one from a group in the US. 2 papers published back to back in Nature Medicine that looked at the brains of Parkinson's patients that had received grafts of embryonic cells from aborted embryos. So this was a clinical trial that was being performed at the time, where they thought, okay, if the problem in Parkinson's disease, at least responsible for the motor problem, is the loss of dopaminergic neurons from the substantia nigra, and it's because of the loss of dopamine innervation in the striatum that patients have the motor complications, why don't we try to introduce dopaminergic neurons in the striatum and hope that these new neurons that we introduce, they will produce dopamine and they will release dopamine where it's not being received anymore. And so at the time, this was way before IPS cell technology and stem cells and all of that. So what researchers could do was to take advantage of abortions that people wanted to do, and they could take the midbrain region of the aborted fetuses, dissect them, take the cells that contain the dopaminergic neurons, and then they did grafts and they injected dopaminergic neurons into the brains of those Parkinson's patients. And there were parallel trials in the US and in Sweden, as I mentioned. And some patients actually benefited from the therapy, others not so much. So the conclusion of the trial was not positive, and that experiment was stopped. But there had been several people throughout the world that had received those grafts. And in the early 2000s and to 2008, that's when they started, those people that received the grafts 12, 16, 20 years before, that's when they eventually died. And then these groups of scientists, they looked at these brains, and what they found was in the regions that had received the grafts, so they could see, okay, this is the region where we injected the neurons. So these are new neurons. They could see, using staining, they could see these are new neurons. These are not part of the normal brain tissue. And they found in these new neurons, or new meaning that they had been there for 12, 16, 20 years. And since they had come from fetuses, they were much younger than the neurons from the 70 or 80-year-old people had in their brains. And they found in those neurons Lewy bodies. And this was very unexpected, but basically it suggested that, okay, these neurons are developing pathology. So is it that the pathology is coming from the old neurons into these new neurons? And this suggested this, together with Brack's hypothesis, it gave support to this idea that there was a spreading, a propagation of pathology in the brain. And then, because of the prion diseases that spread also as disease progresses, researchers made an association and said, oh, this might be a prion-like phenomenon. And what we're seeing is spreading of pathology that is similar to what happens in the prion diseases. And this made the field explode. So now there's a lot of studies on this topic of spreading of pathology. And it seems that this indeed happens. People have been able to model this in mice, where they inject aggregated alpha-synuclein in the brains of mice, and they see that after they inject in one region, weeks and months later, they see signs of pathology in other regions. So this is a very hot topic of research still.
So also they have to be somehow transferred, right? Trafficked, as you mentioned. Trafficking, we mean in cellular biology when proteins are translocated, transported from one location to another. But this is I was not aware of these studies, and I'm curious, what level of inflammation did these people get, because these fetuses are not their own, right? So related or whatever. But now, as you mentioned, the iPSCs, these induced pluripotent stem cells, I think even during the conference in Portugal, there was this lecture about, was it in the context of Parkinson's or Alzheimer's disease? I don't know, but people already try in several labs to get a cell from a patient that has certain phenotype, grow it, reproduce it as a neuron, and try to implant it as a new cell that is not affected. However, if this is a genetic and familiar, right, genetic mutation, then these cells would not be useful, I guess, in the future. Perhaps there should be again some other donor. This is very interesting. It's indeed cutting edge.
You're touching
science.
on important points,
Yes.
and indeed, those patients that received the transplants from embryos, they had to be put on immune suppressants. So that, of course, is not ideal. Now with the IPS cell technology, that you can take some skin cells, reprogram them, and then transform them into dopaminergic neurons. You can reintroduce them into the donor of the cell, so then you don't have the issues of rejection of those cells by the host organism. Of course, you raised the question of, but if the patients have mutations, these are the minority of patients. So only 5 to 10% of patients have known genetic alterations. So it's not that you will find people with mutations in alpha-synuclein very easily. They are rare all over the world. They're very rare. So most people have Parkinson's for reasons we don't know. This is the truth. What we think is, of course, that you need a combination between genetic factors and environmental factors, but we still don't know this very well. We can come back to this later. But there's big hope, and there are trials now ongoing where instead of using these cells from embryos, they are using well-characterized IPS cells that can be injected after or differentiated into dopaminergic neurons, and then hopefully they will perform the function they need to perform in the brain. Whether this will be the solution for the problem, we don't know. There's, of course, many issues there, but there's hope that this could be another possibility as a therapy. Now, of course, with CRISPR and all this technology, even if the cells have a mutation in a gene that we know, it might be possible to correct the mutation and then to introduce the cells from the patient that were corrected so that they don't have the problem. So there's a lot of hope in the field at the moment in this direction, but also in other directions.
It is just fascinating that we are living in such an exciting era, I would say, where biotechnology is developing, science is developing. You have AI, which is helping us also advance. And it's just, I'm so grateful that I am a PhD student right now at this very moment, at this very place. And I'm in this kind of a field of neuroscience, which is endless and extremely complex, but exciting to study.
Exciting.
Right? So if we go back to the spread of these aggregates and in the brain, for example, we don't know how this spreads, but also, so we talked about the origin of the disease in the brain, but there are some hypotheses that, as you mentioned, before you start feeling this kind of tremors, there is some disturbances in your gut, and there are some hypotheses that claim that alpha-synuclein aggregation starts in the gut and is somehow trafficked to the brain. What are these things? Could you please elaborate?
Yeah, no, it's a very interesting topic as well. So, because the pathology was found first in the brain stem and only later in the brain, people started looking. Let's go down. Let's continue looking where we find synuclein accumulation. And they found in the gut, in different tissues, they find Lewy bodies. So then the question is, do they start there or do they start in the brain? And there's now a theory that says you can have cases where it first starts in the body. So it's the body first and then brain later. And then this would be, or these would be cases where the pathology would spread possibly from the gut all the way into the brain through the vagus nerve. This could be one route. And we can come back to this. There's interesting studies there. And there are other cases that seem to start more in the brain and eventually the pathology progresses to the gut. So this, how do researchers study this? Well, they track the symptoms and they see that in some people before they had motor problems, they had already gut problems, and in other people they didn't have any intestinal problems and they had already hyposmia and problems in the brain, and only later they got these problems. This is still an hypothesis because eventually when Parkinson's disease is diagnosed, you already have both. So you need to couple this with certain imaging techniques. So you can image, for example, innervation of the heart, and this is something that is lost in some of the body first cases. So it gives us some ideas that then can be tested in animal models. And in animal models, it seems that you can inject pathology in the gut and eventually see it in the brain, and vice versa. You can also inject in the brain and eventually see it in the gut. So these are studies that suggest that there can be these at least two different forms. And there were some interesting studies, and there's some epidemiological data suggesting that patients that had a vagotomy performed, so they sectioned the vagus nerve because of some other autonomic problems, that this reduces the risk for Parkinson's. It doesn't eliminate, so it means that there's another route, and now people are looking into these possible routes as well. But it seems that the vagus nerve can be a highway through which pathology spreads. How it happens, like you mentioned before, we still don't know. What we know is that you would have to have in some cells in the gut this aggregated protein, and that then this would have to be transported through those intestinal neurons up to the vagus nerve, and then they would have to communicate with neurons in the brain and spread pathology to induce the aggregation of the protein in the brain. How it happens, we still don't know. It's a very interesting problem from the cell biology perspective, because this makes us have to understand how proteins are exocytosed or released from cells, how proteins are endocytosed. There's now this exciting data suggesting that cells may form these structures that can connect to cells, and they are like nanotubes. And people have seen different proteins and mitochondria and lysosomes traffic inside these actin structures between cells, and people have been able to label these proteins, alpha-synuclein and other proteins, and they saw that this protein seems to go from one cell to the next. Now, a lot of this has been done in vitro, because in the human brain it's not easy to image these very tiny structures and to label them. But in principle, it seems that this is possible. And now with cryo-EM and all these tools, people are using more complex models, and they are trying to image these structures. Now, I don't think there will be only one route for proteins to be transported between cells. I think there are several, and maybe one is more responsible for the trafficking than others, but I think several pathways can be involved in the transport of proteins between cells. So we don't know exactly how it happens.
Usually there are these vesicles that transport these proteins, and maybe we can go now in that direction since you also study these processes. Some of these are these microvesicles, some are exosomes. They have different sizes, but in principle they are able to also traffic proteins from one cell to another. Does this play a role in alpha-synuclein aggregation?
Yeah, absolutely. It seems to play a role. Again, what we cannot say is that exosomes or extracellular vesicles are more important than tunneling nanotubes or from other more conventional exocytosis mechanisms. We cannot say what is more important, but we know, we have done work on this and other groups as well, and we can purify exosomes and extracellular vesicles in general, and we can detect alpha-synuclein in these vesicles. Now, we still don't know if it's just because the protein is associating with the membrane from the outside or if it's inside, but we can definitely purify these vesicles and detect alpha-synuclein in these vesicles. So this is established. People believe this is possible, and this opens also a lot of possibilities, because if this is one of the routes, and if we find ways for blocking this particular route, then maybe we can reduce spreading. Maybe we can characterize these vesicles and use tools to understand, is the protein inside the vesicles really aggregated, and can it then transmit this aggregated conformation to other proteins? So there's a lot of open questions, but also a lot of powerful tools that we have available now that enable us to tackle these complicated questions. So, as you said, I think we are living exciting times, and that in the coming years we'll learn a lot that will be important for trying to solve some of these problems.
I've been thinking about the following. So we have these aggregations, they go somewhere, they are trafficked somehow, but it seems, as you described, there are several routes that are almost stereotypical. Something happens in this region, substantia nigra, which is in the brain, then it traffics to another region, and it seems to be always the same parts, the same neurons. Is there anything specific about these cells that allow this trafficking to happen in them, or do we know something about that?
We don't know. It's a good question, and there has to be something that we still don't understand, that is more specific, because we know that in Alzheimer's disease you also have the spreading of tau protein. In Huntington's disease you probably have the spreading of huntingtin protein, and in other diseases people think similar mechanisms may be in place. But the truth is that these different proteins are associated with different diseases. So it's not that proteins will traffic, or different proteins will traffic in exactly the same way and affect all brain regions in the same way. Otherwise you would have just one disease, even if it was caused by different proteins. So that does not seem to be the case. But having said that, we have done some basic cell biology work where we can try to compare three proteins: alpha-synuclein, tau from Alzheimer's disease and frontotemporal dementia, and huntingtin from Huntington's disease. And we put these proteins in simple cell models and try to ask, are these proteins released by the same mechanisms? Are they taken up in the same way? Do they have similar or different functional effects? And we see that they can all be transported or released and taken up by similar mechanisms, but some may be more than others. So there may be some specificity as to what is the primary mechanism for transfer. And then also it seems that the functional consequences, once these proteins are taken up by a receiving neuron, the functional consequences that we studied with multi-electrode arrays just in simple model systems at this stage. But it seems that it can be different. So alpha-synuclein can have slightly different effects from tau, and tau can have different effects from huntingtin. So it depends a bit on the protein, how the receiving cells will respond to the accumulation of these proteins. So that's something we need to know more. And again, this is difficult to do because we're talking about events that are not happening at high rates, and we're talking about very small amounts of proteins that we have to manipulate, and then we have to be able to measure effects or to use imaging approaches to see where the proteins go. So there's a lot of challenges, but again, I think we're starting to have tools that will enable us to get some answers.
Yeah, and what is also a variable that is hard to control is the time, right? This takes a lot of time to go from one place to another, and it's fascinating. It's indeed fascinating. So we talked about these dopaminergic neurons. These are neurons that produce dopamine, one of the neurotransmitters in the brain, right? And these aggregates, they affect neurons, but also in the brain, as we know, there are other cell types, the glia. Then we have the astrocytes, the oligodendrocytes, and the microglia predominantly, right? How important are they in this process of trafficking or aggregation, or how do they contribute to the pathology of the disease?
No, absolutely. This is a very important question, and the strongest evidence we have so far comes from Alzheimer's disease, because in Alzheimer's there are some risk factors that directly implicate, for example, microglia in disease, and people carry some mutations in some genes, and these are microglial genes that regulate inflammation by these cells. So now we are paying a lot of attention to this also in Parkinson's, in other diseases. And it seems that, microglia for example, but astrocytes and oligodendrocytes will also play important roles. But microglia can actually regulate important neuronal functions in simple cell biology, such as autophagy. So there was a very recent publication in Neuron that we actually discussed today in our journal club that shows that in Huntington's disease models, you can have microglial cells secrete chemokines that will then communicate with some receptors in neuronal cells and alter autophagy in neurons. So this means that these microglia from people with disease, they may impair autophagy in neurons, and this could then lead to the accumulation of more proteins in neurons that make them dysfunctional. So we still don't know, is inflammation good or bad? And now we think there's good inflammation and bad inflammation. So nothing is simple in biology. When you think, is it good or bad? There's not a single answer. It can be good, can be bad. It seems that depending on the type of inflammatory response you have, this can either aggravate disease, or it could be also a beneficial response that tries to modulate the system so that the system can fight the problems. But it's important, as you said, to understand what microglia, or what is the role of microglia. Astrocytes are, of course, also considered to be part of this tripartite synapse where you have two neurons communicating, and then astrocytes also releasing factors and taking up factors from the synapse. So I think we'll need to really increase the complexity of the models we use. And of course, it's nice to isolate questions and to try to get simple answers, but at some point we will need to increase the complexity. And now, again, we are living times where we can use organoids, which are three-dimensional culture systems where you can have multiple cell types. Of course, they are not perfect because there's still some cell types you cannot have in there. Microglia are not from neuronal origin, so they come from outside. There's problems with vascularization of organoids, and so there's issues. But again, I think it's exciting that we're having tools and access to models that may enable us to address these more complicated questions.
If you want to learn more about organoids, you can go to our previous podcast with Dr. Outeiro, where we talk about them and iPSCs.
Patapi is an expert.
Yeah. Exactly. I even have some organoids in the lab, and I stain them for my protein of interest from the extracellular matrix, and I'm curious what is going to be the outcome. Maybe this week I will find out. Before we continue with Parkinson's and disease-related topics, I think now is a great moment to talk about, again, animal models. You just said that we have to increase the complexity of the models, and this is maybe one of the reasons why we need higher animals to study these diseases. And now, in particular in Europe, there is a huge debate whether we have to shut down research or not. And we want to spread the word and say that we need animal models in order to progress in science and humanity as well. So maybe couple of sentences. Why do we need this complex, and how do we study Parkinson's and other neurodegenerative diseases despite the lack of these phenotypes, parts of some of the phenotypes that we see?
I want to be very clear. We as scientists, we need to have the sense of responsibility and try to minimize animal suffering as much as possible. That should be a priority. I want to be very clear about that. We should not use animals just for the sake of using animals. There has to be a good question and a good reason for using animals. Now, we also discussed these diseases are very complex, as I hope it's becoming evident from our discussion. And there's many aspects that cannot be addressed using simple model systems. We now have, as we also talked about, and you discussed this in the previous podcast, we have these cell culture systems, 2D IPS cell cultures, or 3D organoids, brain organoids. So there's a lot of hope that eventually we will be able to have these culture systems without requiring the use of animals and to minimize their suffering. But there are certain aspects of research in this field and in many fields that can only be tackled by using models that recapitulate the complexity of the biological system. And one can say, yeah, but we've used animals for decades and we still don't have a cure for Alzheimer's or for Parkinson's. That's true. But that does not mean that those models are not useful. And they are useful. They have to be used responsibly. We have to ask the right questions, but we cannot hope to cure these diseases without testing some of these hypotheses we are testing in a real living organism. And so mice and rats are very useful because we have a lot of knowledge and tools, and they live less time, so things can be done more rapidly. Of course, that on the other hand, that also poses limitations because maybe they don't mimic all the aspects of the disease. Non-human primates are very useful, and they are being used all around the world, in China and in other countries that are not as strict with these laws. So I think it's really a mistake for Europe to be so influenced by those that are defending the animal rights, and nothing against them. Everyone should be able to express their ideas in a democratic manner with respect, without creating problems and disruptions. But they should be able to express their ideas. What I think is a problem is that we as scientists tend not to be as vocal and tend not to come and defend the need for using these types of models. And I think, as I said, we want to use them responsibly, but in order for us to be able to stay competitive in this research field and in many others, we cannot simply ban the use of animal research. I don't know if in 50 years or 100 years we'll be in another position where we can say, now our models are so good that we don't need them anymore. But this is not where we are today. And of course, we should make efforts towards that goal of developing models that will be as good or better than animal models. But a lot of people are working on that. There's many groups in the world trying to develop that field. In parallel, we still need to use animal models. And I have to say, this has been very frustrating here in Germany. We all know in Göttingen, but talking with colleagues in other parts of Germany, they're facing the same problems. It's unacceptable. This is a word that I use sometimes because, we apply for a license to be able to do some experiments in animals, and it takes more than a year, sometimes 2 years. And we go back and forth with the veterinarian authorities, and it seems that we are not making progress. We have situations where we got funding from a company to do a certain project in animals. It took us more than 2 years just to be able to start the experiment. And at some point, you can face a situation where the research you proposed that was eventually approved by the authorities is not valid anymore. So you don't even want to do it. And then you run into problems, because then if you don't do what you propose and what was approved, then the authorities come and you get into trouble because you didn't do what you proposed. But I mean, after 2 years or 3 years, things change. So it's a catch-22 situation. I think authorities are not serving the human society well just by trying to block animal use. What they should do is help and work with researchers in order to enable us to speed things up, to comply with the 3 R's rule, with this rule that everyone accepts, to reduce, to replace, to refine the models. But we need the models. This is the bottom line. And I want to send a very clear message that I understand that we should not do research just to induce suffering in animals, but we need to use animals because nothing recapitulates the complexity of the human disease as an animal. And so we still need it.
Thank you very much for this wonderful summary. I'm very happy that you really touched upon these topics, and I hope we can reach more people that are not familiar. Why do we need animal models in our research? And hopefully we can contribute somehow to this debate and kind of make the system working a little bit better. I also suffer personally from this, waiting for a license to use a certain tool. And as you know, PhDs are limited, and also postdocs positions are limited. And this is also limiting our work, not only as a person, but as a lab. And then in this country, you become to be not that competitive, although you have the capacity in terms of people, in terms of facilities. However, in certain places in the world, in the US, maybe even China, although let's not talk about this part of the world right now, but you have this kind of more developed system that facilitates or helps scientists to do their job in a faster, more efficient way. But we kind of deviated from the topic of Parkinson. I would come back to this in our last bit of our episode, where we discuss problems in science. Let's go back to Parkinson's. You said that we don't know what is causing it, more or less. We have some idea. However, what are the treatments of people with Parkinson's disease? And is there anything that we can do in our daily life to prevent ourselves from getting this disease?
Very good questions. So the therapies for Parkinson's disease have not changed much for the past 60 years. The best treatment we have today is levodopa. It's a precursor of dopamine that patients can take as pills or as different forms, that gets converted into dopamine in the brain, and that basically replaces the dopamine that the neurons are not producing. This is the gold standard. This is what really helps the patients. And especially in the beginning, in the initial years of disease, this works very well. So patients live what's called the honeymoon phase, where levodopa is working great, and they almost forget that they have Parkinson's. After a few years, because there is continuous disease progression and continuous cell loss, other symptoms appear, and then things get a bit more complicated. Fortunately, because we understand the dopaminergic circuitry relatively well, we know and we have some molecules that we can use to fine-tune the function of this circuitry in the brain. Eventually, things also stop working or don't work as efficiently. Dyskinesias, these involuntary movements can appear, and therefore other more advanced therapies can be used. They depend on the patient, so that it's not that one therapy fits all. This is also important because people sometimes compare, but my neighbor had this therapy and I should do it as well. This is not how it works. This is it's a disease that requires that the patient forms a good team with their movement disorder specialist, with their family, that everyone pays attention to what are the biggest challenges that the patient faces, and then with the clinician they can decide, in your case, for your particular problems, maybe this option is the best or that option is the best, and what are these other options. So one that is performed in many countries throughout the world and that helps a lot of people is deep brain stimulation. So this is a surgical procedure that basically implants some electrodes, some thin wires into the brains of the patients in regions where the electrical stimulation that is not happening normally because the dopamine is not there, but these wires, they can deliver electrical stimuli that make the circuitry work better. And then the patients have an implant under the skin of a device with a battery, and that can be programmed to deliver more or less stimuli in different directions in the brain. And that works very well for some patients, also for some time. In other patients, this is not an option because either their disease is too advanced, or they have signs of dementia, and then this cannot be performed. Then there's other options. There's pumps that inject dopamine into the stomach, the duodenum, and it delivers dopamine there in a continuous manner to avoid the fluctuation. So one problem that happens eventually is that patients have periods where they have peaks of dopamine and peaks of less dopamine, and they have what's called on and off periods. So there's fluctuation. So they know everything is working well, and at some point they have an off period and they feel frozen. So then they need to take levodopa in order to be on again. And these fluctuations are for reasons we don't fully understand, but some have to do also with the gastrointestinal problems. So the way the contractions work in our GI tract work, sometimes this is not working well, and the medication is not progressing and reaching the area of the gut where it's absorbed. And so in some cases, it's better to have this pump that delivers the medication continuously. Now there's new formulations like a sublingual, it's not a pill, but it's like this mint things that we put under the tongue. So it's something similar that can be taken more rapidly. So if the patient feels like they're going to have an off period, they can put this under their tongue and let it dissolve, and it will release the molecules there, and that will help the patient. So fortunately, there's different formulations that can help patients. Now there's also another technology that is still a bit controversial, but it helps some patients. That's called focused ultrasound. And what this technology does is to use ultrasound to damage some parts of the dopaminergic circuitry that are sending certain signals that need to be reduced. So we have inhibitory and excitatory regions of the circuitry. So with focused ultrasound, you can damage and reduce the activity of the inhibitory regions, and then the circuitry works because you're not inhibiting as much. And this works. It's not performed everywhere, but there are some centers in the world that are specialized in this type of therapy.
How invasive is this therapy?
It's not invasive. It's a procedure. I mean, you have to have the head immobilized with a frame, and you have to have very good imaging to know exactly where you're gonna hit with the ultrasound. But it's not a surgical procedure, so there's no cutting of the skull or anything. Whereas in deep brain stimulation, you need to cut the skull, you need to drill a hole into the skull to introduce the electrode. So it's a more invasive procedure. But so these are some options, therapeutic options we have at the moment. But of course, we know that none of these is stopping disease from progressing or reverting disease progression. So that's the goal. In the field, what we would like to have is a strategy that stops disease from progressing from the point when it's diagnosed, or that we are able to diagnose it even before the motor symptoms take place, to prevent it from happening altogether. And now, again, exciting times we're living. There's a recent biomarker that was published. The technology has been published already a couple of years ago, but there was a recent study suggesting that this really works. You can use a biomarker from the CSF, and you can measure some properties of the alpha-synuclein protein, and this can tell you if you have Parkinson's or not, or other synucleinopathies. These diseases also have problems with alpha-synuclein. And this is going to be very useful, not necessarily because we'll be able to treat the patients, but because we'll be able to measure this and to assess the effects of therapies. So to see if we are having any beneficial effect at this level of this particular biomarker. So there's a lot of exciting stuff to happen in this field in the coming years as well.
Yeah, so under biomarker, you mean these molecules that can be used to identify if this disease will happen, and what will be the type of the disease, etc. And CSF is the cerebrospinal fluid. I just want to clarify this term, since we use it. This is a great point to ask you the following question. I stumbled upon an article of a person who has hyperosmia, I think it's called, where you have this super sensitive smell. And these people, or this particular person, claims to be able to smell people who will suffer Parkinson's disease in the future. And I think there are studies now going on with this person in Great Britain. You're definitely aware, and you can tell us more about this. How is this possible at all?
No, that's very interesting. So it seems that indeed there was a lady that her partner had Parkinson's disease, and so she was used to this smell. And one time she felt in another person a smell that she recognized from her husband, and this smells like my husband. So they started looking into this, and they found that she really has this ability of distinguishing the scent of the skin of a Parkinson patient. Dogs can also do that, and so what we know is that indeed the sweat in Parkinson's patients seem to be altered. These people have some special molecules in their skin that have a special smell, and normal humans cannot pick it up, but it seems that some people with or some animals might be able to pick it up. And there was a study also done recently on this, where people tried to use very sensitive techniques like mass spectrometry to measure what could be the molecules that are characteristic in the skin of Parkinson's patients. And this points us in one direction that is also emerging a lot in the field, that this may have to do with lipids. So the disease, we tend to think about this disease as a proteinopathy, so problems with proteins, but we know that the cell biology changes in a way that also leads to changes in lipid metabolism. And we just published a paper, it just came out a couple of days ago, really discussing this idea that in addition to being proteinopathies, these diseases are also lipidopathies. And this is something we need to understand in greater detail, because instead of only trying to interfere with the protein aggregation, maybe we can use other types of molecules that interfere with lipid metabolism, and maybe that's also beneficial in Parkinson's disease. We'll have to see.
I think this would be a great topic for our future meetings. I would be keen on learning more about this topic, and maybe in some time in the future we can meet again and we can discuss only this particular area or paper. But I wanted to ask you the following question. So let's say we are able to detect Parkinson's 20 years in advance, 30 years in advance. Can we do something to prevent it from happening?
Yeah, sorry, I remember you were asking about that as well. So what can we do to prevent? We don't know. This is the simple answer is we still don't know. But we now know that, for example, exercise really seems to slow down Parkinson's. We cannot say, it prevents Parkinson's, because there's many examples of athletes that develop Parkinson's. And I'm not even talking about Muhammad Ali, because he was a special type of athlete. And we know that in people that have head trauma, the risk for Parkinson's and for other diseases increases. So I'm not talking about that type of athletes, but other types of athletes may also develop Parkinson's. So having said that, we know exercise there's now evidence showing that it can slow down progression. So there's experts, clinicians that are experts in this field that are recommending their patients to have their at least 10,000 steps per day and to be active and to exercise whatever form of exercise, as long as it increases your cardiovascular rate, it seems to be beneficial. It doesn't prevent it, but it certainly helps. Nutrition is another field that is growing tremendously a lot, and we know that this may also influence the progression of the disease. How exactly, we don't know, but of course there's Mediterranean diet and things we know that we should avoid or include in our diets that seem to be beneficial, like coffee drinking, moderate. Of course, we don't want someone to, have an overdose of caffeine, but that seems to also be somewhat protective. And then there's other antioxidant-rich foods that may also play a role. The problem is that it's very difficult to find associations between these types of human behaviors with disease. But there's anecdotal evidence supporting a lot of these as possible disease-modifying strategies. And I think we'll also learn more about this in the coming years. Now that we are focusing on exercise and nutrition, more studies will be done to try to isolate the factors that are really affecting disease progression. And so those two for sure are something I would say, if we do that, if we include those in our lives, we may have a chance to delay disease.
What about social contacts? We heard last time that next to exercise, diet, social contacts are quite important for whatever reason.
Absolutely. So this is, again, it's one of these recommendations that people always make. And I think, it's easy to explain to our listeners. So social contacts are important because by talking to people, by going out, by traveling, by dancing, by listening to music, we will be stimulating different areas of the brain. And we know that a lot of these diseases are not only due to cell loss, to neuronal death in the brain, but also to loss of synapses. And how can we try to keep our synapses active and to keep our cells connected? By stimulating them. And it's not just playing Sudoku the whole day that will do it, right? That may be one part of it, but then having social interactions, traveling, dancing, going out, all of these activities that involve contact with other human beings will probably help our neurons keep better connected. And I think in that sense, they can be valid and important.
And also, of course, sleep is very important in general, not only, but sleep also, we have our neurons fire again, they maybe enhance or not these connections, and it's important in many, many, many aspects. But in general, we have this always coming up, this exercise, diet, sleep, social contacts, that is kind of a universal recipe of healthy lifestyle, which I like and I try to follow, right? So I think we summarized to a good extent Parkinson, how it develops, how do we treat it, how can we maybe prevent it, and we talked about very exciting topics. I would like to change the focus now, since we are kind of running out of time slowly, and would like to go back to the problems of science. In science, we already touched upon this animal a problem with the animals in research. However, we have many other things to deal with as a scientist. For example, some studies are hard to be reproduced. There is this reproducibility problem. Also, many people that are trained as a PhD students, they leave academia, which per se is not a problem, but academia is suffering from highly qualified people. There are also troubles in terms of funding, publishing, and many aspects that we need as a community, I believe, to work on in order to improve our work. What would you think is, for you personally, one of the most bothering problems of this that I described, or maybe you suggest something different.
No, I think this is true. In academia, we find a lot of these problems you mentioned, and we can talk about each of them individually. I think they end up all being connected in some way or another. And, usually academics are people that have, this curiosity, this interest in learning and finding answers to questions and problems for which we still don't have the level of understanding we would like to have. And because we're curious, we want to do the best we can. A lot of people in the field have a special drive, a special ambition that they want to be the first to make a discovery. We know that funding is very difficult, as you said. The funding rates all over the world are much lower today than they were 30 years ago. And with the problems the world is facing today, I don't think this is going to improve in the coming years, because we are spending more in military and energy and other problems. So there won't be as much money for research, unfortunately, because I think research is what can drive the humanity further. And that was seen when there were big bumps in financing, in public spending. In science, that's when men went to the moon and when big achievements were done, or were made. So I think we need to have this ambition to solve big questions. Now, because there's not as much funding, this creates pressure in the system. Everyone is competing for the same pots of money. And this sometimes, in some people, may pressure them to cut corners, because they know that they need to get funding to be able to do the work, to get their secure positions, and to publish papers. So this is, again, a vicious circle that puts some people that may not have the highest ethical standards into more pressure to cut corners and to eventually publish things that are not reproducible. So I'm touching very superficially on several of the ideas that you brought up. So I think, we need to train the younger people so that they understand science is not easy. It's not a 9-to-5 job. I always tell this. If you want a 9-to-5 job, please don't come into science, because you will be unhappy, you will be miserable at times. You will not be able to balance your life properly, and this will lead to mental health problems that, of course, we know is a big thing everywhere at the moment. So science is hard. It requires a lot of commitment and dedication. You need to be passionate about what you do. Try to find a project, try to find questions for which you are passionate about. You need to have the curiosity. You need to want to know everything that is being done. You need to want to read all the papers that are coming out in this field. You need to know more than your supervisor. Don't expect that, I don't know this, I'm going to go and ask my supervisor. I tell my students, you should know more than I do about the specific topic you're working on, because you need to be reading all the papers. And so I think this message is very important. If you think that you want to have a 9-to-5 job, nothing wrong with that. Everyone should be happy doing what makes them happy. But this is not science. If you're passionate, then try to be the best you can. And bring ethics with you, your ethics, and know that there's cutting corners and changing findings is never the way. This is something that should never take place. You should always do your best to tackle the questions you're tackling. Of course, we can disagree on the interpretation of data, but all the interpretations should be backed up by real data. And I think this is an idea that is very important. Now, I don't think this will explain all the issues with reproducibility. And I think this has to do with the complexity of biology. Biology is not just black and white. There's a million shades of gray in between. And many times we don't understand really all the variables that make me obtain a result in my lab. And then in Japan, colleagues thinking that they are reproducing what I did may find different results. And it's not that I'm wrong and they are right, or vice versa. I think the issue is that sometimes we don't understand all the complexity of biology and we may run into discrepancies. So how do we deal with this? We have to be as rigorous as possible in writing and describing what we are doing so that others can try to really mimic all the conditions we used in our experiments. Now, the system has many other problems. I think, we, because of the competition and the pressure to obtain funding and publish papers, we are put in a situation where many things are working poorly in the field, and we are all asked to review papers by other colleagues. The peer review process is a problem. We don't know if there is a better system. People are trying to integrate different ideas in using double-blind peer review. It seems that that's not going to help, because it's easy to know who are the groups working on a certain topic, and because people tend to cite their own data. And if you write, as we previously shown, then you don't need to have your name, but the reviewer will know from where the study is coming from. So I don't know. I don't have the solution for this. But what I know is that we cannot use we should find alternatives to the metrics we're currently using for assessing the quality of scientists. We need to give early career researchers an opportunity to enter the system. We need to help them get their funding and publish and get recognized for what they do, but also without pressuring them to just enter the system without a network of support. I think it's better sometimes to keep them in the system and give them an opportunity to grow gradually than to just launch them into the fire. I think that's not necessarily helpful. But still, in this publication business, I think the whole business is wrong. I mean, we have a business that is excellent for the publishers, but for everyone else, this is terrible. I mean, they publish, they sell the journal, they get the publicity from the sponsors they get. You pay to publish in their journals, and it's not a small amount, as you know. Some journals charge 10 and more thousand euros for publishing a paper, and so you pay a significant amount, and you pay from grants that the government gives you. So instead of using 10,000 euros for research, you're now having to pay 10,000 to publish a paper, so that this gets the visibility that will maybe enable you to get more money. So it's crazy. Then we do the work for the journals because we review for free. So they ask us, can you review this study that we received? And we many times say yes, of course, because this is collegiality in the academic system. I review because other colleagues will review my work, and it used to be like this in the past. But now, because there's so much pressure, so much competition, I try not to do that, but I cannot say that I'm always unbiased. I try, but maybe I'm not always as unbiased as I should be. But it's not uncommon for all of us to have experienced situations where the comments from the reviewers are just destroying our work. They are not being helpful and trying to suggest things that we could do to improve the work. They come and say, this work is bad, it's not an important contribution, this should not be published in this journal. And I always think, when I'm asked to review a paper, I think, first of all, it's not my decision as a reviewer to say whether this study is for that journal or not. If the journal asked me to review, it's because internally, the people that are responsible for the journal thought perhaps this fits that journal. So I think it's not my role to tell the journal, this is not for you. But many people are very opinionated about that. And they'll say, this is not for this particular journal. I think that's wrong. And then they are just finding the problems. and they're, oh, this is bad, this is not right, this is that, but they don't give the suggestions, the constructive suggestions that will help the authors improve their study. And that should always be the point of review. So in peer review processes of papers, these are several of the problems I identify. In grants, the problem is even more dramatic, because there you have one shot, basically. You submit a grant application. If you are unlucky to have a reviewer that is not 100% positive, this will decrease your chances of getting funding, because it's so competitive that if you don't make sure that all the reviewers match in their opinion and support, then your chances of getting that project funded decrease significantly. And we all know what this means. Many times, you submit one project, you wait 6, 7, 8, 9 months, 12 months for the answer. And only if everything goes well, maybe 1 year later, you get the funding to do the project. This is delaying progress. It's delaying science. And many times, as we also know, the project doesn't even get funded. So all the work you put into writing a project that you thought was valid at that time ends up never getting funded, never seeing the light of day. So you wasted a year trying to propose something that will never get funded. And then you have to go to the next thing, because time passed and what you proposed 1 year before may not be current anymore. So you have to go to the next thing. So it's a very difficult system. There's many problems. I don't know what the solutions are to all of these issues, but I think this would require also the scientific community to really work together and discuss these things, not just superficially and say, no, we cannot use impact factors, or we cannot do this and that. There's a lot of talk, but I see little action. I wrote some years ago a paper. I was frustrated, I have to say, at the time with some decisions I thought were unfortunate that I faced, and I'm sure everyone faces them. And I wrote a lot about this and how I think that the system needs to be shaken. And the people that can do this are the people that are in the higher positions at the moment. Of course, it's not a student or a postdoc that is in a position to really shake the system and make it change, but the people that are higher up, that have gone through the ranks and made it to the top, and those that have the experience and that know the problems, I think those are the ones that have more responsibility to try to influence the system and change it. And I think with journals, I think we should go on strike. We should just not review for the journals. I mean, why should we work for free for them and still pay and thank them very much when they publish our papers? I mean, this is a difficult situation. It's a game we all play, but to be honest, I think this game is not correct.
It is a huge scam. And at the end, taxpayers are the ones that actually fund science and then pay to this kind of institutions. Recently, I participated in an evaluation of a SFB grant. This is a big amount of money that is awarded to a certain group of scientists in a certain amount of years. And I was surprised. I mean, not that surprised because I kind of know, but how much politics is involved in every step of this process. And it's just insane that I talked to some of the more advanced scientists about this process in other countries and what did they experience. And I heard about stories how these big decisions are already pre-made, although you kind of compete and you put, as you said, a lot of effort, years in this design of your project and about the funding, etc. And at the end, it is so wrong. But you said people who are at the top, they have to kind of try to shake the system. One problem that I see is that these people are not by chance at the top, and maybe they feel very comfortable there, right?
Yeah.
And I think that also we as a young scientist, we cannot have this influence, but we have to start speaking and somehow trying to think about other systems that will be more beneficial for all of us. And I think this is our responsibility as young PhD scientists. So thank you very much for raising also this question. I think it's very, very important, and I always talk to my peer scientists on my age about this, and we kind of agree, and we know that we have to do something, but we kind of don't know. We work, as you know, on our projects and etc. However, this was a very, very, very good summary, and I think it's a very good point to stop our discussion here with a lot of open topics for the future discussions, hopefully. And once again, I would like to warmly thank you for your time and for your nice overview on so many interesting topics. Thank you very much for coming.
No, it was a pleasure. I really enjoyed, and I'll be happy to come back whenever you want to discuss any of these issues in the future. So I hope this was useful for everyone watching us, and thank you very much.
Great. Neuroscience and beyond. Know more, get inspired.